In 1995, scientists counted every single kakapo left on Earth. The number was 51. That was the entire species. And every expert looking at those numbers said the same thing quietly to themselves, in their reports, in their labs. This bird is not going to make it. The genetics alone were catastrophic. The population had crashed so hard and so fast that the surviving birds were essentially all cousins. Inbreeding was rampant. Egg infertility was through the roof. Conservation science had a rough rule. You need at least 50 genetically unrelated individuals to have any real shot at pulling a species back. The kakapo had 51 total birds, many of whom were closely related. But here is what nobody knew in 1995. The kakapo had already survived something far worse than this, long before humans ever arrived, and the DNA told the whole story. First, you need to understand what kind of creature we are talking about, because the kakapo is not just rare, it is genuinely one of the strangest animals that has ever existed. It is a parrot, but it cannot fly. It is the only flightless parrot in the world. It is also the heaviest parrot on Earth, with males reaching almost 9 pounds. It has an owl-like face with forward-facing eyes and a disc of feathers that funnel sound toward its ears. It is entirely nocturnal, and it can live to be 100 years old. There are kakapos alive right now that may have hatched before the Second World War. Its entire reproductive cycle is locked to a single tree. The rimu tree, a native of New Zealand, produces a burst of fruit only once every 2 to 5 years. That event is called a mast year. Until the rimu masts, kakapo females will not enter breeding condition. The tree decides when the species reproduces. When it does, males dig bowl-shaped depressions in the earth to amplify a deep, booming call that can travel 5 kilometers through the forest. And they do this every night for months. At the end of all that effort, a female might or might not show up. This is a creature that evolved in a world without ground predators. New Zealand had no native land mammals. The kakapo's only real threat came from above, the now extinct Haast's eagle. So it evolved a freeze response. When threatened, it stops moving. Its moss-green feathers make it invisible against the forest floor. Then, humans arrived. First the Maori around 700 years ago, then Europeans in the 1800s. With them came rats, cats, and stoats, ground-hunting predators that used their nose, not their eyes. And the kakapos stood completely still while they were killed. The freeze response that had protected them for millions of years became the thing that ended them. By the 1950s, kakapos survived only in the most remote corners of Fiordland on the New Zealand mainland. By the 1970s, searchers could find only males there, no females. The species was declared functionally extinct on the mainland. Then, in 1977, on a large island called Stewart Island, 30 kilometers south of the mainland, researchers discovered something extraordinary. A breeding population, around 200 birds, including females. The species was not dead yet, but Stewart Island had feral cats, and the population was declining fast. Through the 1980s and 1990s, conservationists moved every kakapo they could find to small, predator-free offshore islands, scrubbed those islands clean of every introduced mammal, and watched the birds constantly around the clock. By 1995, that effort had landed on a number: 51. Before the mainland population collapsed entirely, wildlife officers made one more discovery. Between 1974 and 1977, they found 18 surviving male kakapo in Fiordland. All male. No females. One by one, most died. But one survived long enough to matter. One male was captured and transferred to the predator-free islands. He was named Richard Henry, after the 19th century Irish conservationist who first proposed using offshore islands as sanctuaries for New Zealand birds. Richard Henry was the last kakapo with Fiordland genetics. the last representative of the mainland lineage. The Stewart Island birds and the Fiordland birds had been separated for thousands of years, and their DNA had quietly diverged across all that time. For decades, Richard Henry lived on the islands and did not breed. Then, in 1998, he mated with a female named Flossie. 3 chicks hatched: Kuya, Gulliver, and Sinbad. 3 birds. That was the entire legacy of Fiordland mainland genetics in the modern world. Those 3 chicks became the most genetically valuable animals on the planet, their father's lineage carrying approximately 1/5 of all unique genetic alleles not found anywhere else in the kakapo population. Richard Henry died in 2010, estimated to be over 80 years old. His son Gulliver is believed to carry unique immune system genes called MHC alleles, found in no other living kakapo. As you will hear shortly, those genes may be the key to the species surviving its newest invisible threat. But first, the miracle in the DNA. In 2021, researchers from Stockholm University, the Swedish Museum of Natural History, and the University of Otago published a paper in the journal Cell Genomics. They had sequenced 49 kakapo genomes, 35 from the living Stewart Island birds, 14 from museum specimens of the now extinct mainland population. What they found changed everything. The mainland birds, the ones that went extinct, had an average of 34 harmful mutations per bird. The surviving Stewart Island birds had an average of 18, roughly half. The birds that survived had fewer genetic defects than the birds that went extinct. Not more, fewer. This is the opposite of what conservation science predicted. Small inbred populations are supposed to accumulate harmful mutations. The theory said that when a population shrinks, random chance causes mutations to build up faster than natural selection can eliminate them. The smaller the population, the worse the genetic load. It is called mutational meltdown, and it was supposed to explain why small populations spiral toward extinction. The kakapo broke that model entirely. The explanation lies in 10,000 years of island isolation. When Stewart Island separated from mainland New Zealand as sea levels rose at the end of the last ice age, the kakapo there were cut off completely. For thousands of generations, they bred within that closed group. Constant inbreeding exposes hidden mutations. In a large, genetically diverse population, a harmful recessive gene can hide for generations behind a healthy copy. Most individuals carry only one copy of the bad gene and never show symptoms. But in a small, inbred population, the odds of inheriting 2 copies of the same bad gene skyrocket. When that happens, the damage becomes visible. If severe enough, those individuals do not survive to reproduce. The mutation is removed from the population. This process is called purging. And the kakapo had been doing it involuntarily, relentlessly, for 10,000 years. Dr. Nicholas Dusex of the Center for Paleogenetics at Stockholm University put it this way. The surviving Stewart Island population had been isolated for approximately 10,000 years, and during that time, harmful mutations were removed by natural selection through purging, facilitated by inbreeding itself. His co-author, Professor Love Dalen, added, "The Stewart Island kakapo have been suffering from inbreeding for 10,000 years, and consequently, when the population shrank further in recent centuries, they were likely less affected than they would have been without going through this earlier purging." Think about what that means. The very thing that was supposed to doom them, the isolation, the inbreeding, the small population size, was quietly building a genetic resilience that the more diverse mainland population never developed. The mainland birds hid more damage behind more variety. When the mainland population collapsed, the hidden mutations came out. The island birds had already paid their price across 10,000 years. And when their numbers crashed to 51, their DNA was in better shape than anyone knew. This does not mean inbreeding is harmless. Since 1985, 40% of all kakapo eggs laid have been infertile, and another 20% of embryos die before hatching, 3 to 4 times the rate seen in healthy parrot species. But the purging discovery meant the team had more to work with than the numbers suggested. The birds were not genetically doomed, they were genetically battered but functional. And with the right management, that was enough. In 2015, the Department of Conservation, working with the University of Otago and Duke University, launched the Kakapo 125+ project. The goal was unprecedented: sequence the complete genome of every living kakapo on Earth. A crowdfunding campaign helped fund it. By late 2018, every adult had been fully sequenced. For the first time in conservation history, a wild species had the complete genome of every living member on file. This enabled a revolution in management. Scientists built a complete genetic family tree of the species. Every bird's relatedness to every other bird is precisely known. Pairings are chosen by algorithm to maximize genetic diversity in the next generation. Genetic signatures now predict which males carry poor sperm quality, which females are at higher risk for infertile eggs, and which birds may need targeted veterinary attention. During breeding seasons, every food hopper on the island is coded to each bird's transmitter signal. A hopper only opens for the specific bird assigned to it, automatically records their weight, and the team adjusts each individual's diet based on that data. Every kakapo is on a precision nutrition plan. Which brings us to one of the strangest and least known facts about this entire story. Kakapo females can control the sex of their offspring. Male kakapos grow 30 to 40% larger than females, requiring far more nutritional investment from the mother. When a female is well nourished and her body condition is high, she produces significantly more male chicks. When she is in poorer condition, she shifts toward producing females. A large dominant male in a lek system can father dozens of offspring. A smaller female is a safer investment when resources are tight. This is the Trivers-Willard hypothesis in evolutionary biology, and the kakapo demonstrates it clearly. But supplementary feeding programs in the 1980s accidentally created a crisis. It worked for breeding, but 70% of chicks born were male. In a species that breeds every few years, that is a slow-motion catastrophe. The fix was counterintuitive. Scientists deliberately underfed the better-conditioned females, brought their body weight down, and shifted them toward producing daughters. It worked. This was the first time sex ratios in an endangered species were deliberately altered through maternal nutrition, published in the journal Nature. Now, 2019. Because 2019 was the year everything went right, and then something went terribly wrong. And then the science saved it again. The Rimu trees produced a megamast that year. The biggest fruiting event in decades. Over 200 eggs were laid. 72 chicks fledged, the best breeding season in the recorded history of the species. And in the middle of it, birds started dying. A fungal disease called aspergillosis swept through the population on Wenuahu, also known as Codfish Island. It is caused by a common environmental fungus that produces severe respiratory illness. In the previous decades, only one kakapo had ever died of this disease. In 2019, 21 birds were infected. 9 of them died, 2 adults and 7 chicks, approximately 4% of the world population gone in a single season. The genomics team was called in. They sequenced the genomes of the Aspergillus fungal strains found in every infected bird and in environmental samples across the island. What they found was extraordinary. Every single infected bird on the island had been infected by the exact same fungal clone. One strain, one source. The most likely explanation was that humans had introduced it through contaminated equipment or materials moved between nests. The research also revealed a climate warning. The warm, dry conditions that created the megamast year also created perfect conditions for Aspergillus. As climate change makes such seasons more frequent, that fungal pressure will increase, which is a serious long-term threat for a species with a compromised immune system. This is where Richard Henry's son Gulliver becomes important again. Gulliver is believed to carry unique MHC alleles, immune system genes, found in no other living kakapo. Some researchers believe that selectively breeding Gulliver's line may be key to producing future kakapo capable of withstanding aspergillosis. One bird, decades after his father was the last of his kind, still carrying something the rest of the species lost. And then there is Sirocco, a kakapo who did not save the species through his genetics, but through his personality. Sirocco hatched in 1997, fell ill at 3 weeks old, and had to be hand-reared. This caused him to imprint on humans rather than on other kakapo. In 2009, during filming of a BBC documentary with Stephen Fry and zoologist Mark Carwardine, Sirocco climbed onto Carwardine's head and attempted to mate with it at length. The clip has been viewed over 30 million times. New Zealand's Prime Minister appointed Sirocco the official spokesbird for conservation. Attempts to collect his semen led to the creation of the ejaculation helmet, a rugby helmet lined with dimpled latex. It failed. It now sits in the Te Papa Museum in Wellington. But Sirocco's fame brought millions in donations and introduced new audiences to kakapo conservation worldwide. The technology supporting the living birds continued to evolve. Three-dimensional printed eggs, made by Weta Workshop, the company behind Lord of the Rings props, are placed in nests to emit lifelike chirping sounds that prepare mothers for the moment of hatching. Salmon from genetically valuable males is transported between islands by a drone the team nicknamed the Cloaca Courier. In 2019, artificial insemination raised egg fertility from 29% to 70% in second clutches. 4 chicks that season were confirmed born through artificial insemination, including offspring from 2 males who had never reproduced, one of whom carried Fiordland heritage. As of 2026, approximately 235 kakapo are alive on Earth. Everyone is named, everyone wears a transmitter, everyone has their complete genome on file. And right now, 2026 is a Rimu-mast year. The trees are fruiting, new kakapo are being born for the first time in 4 years. What started as 51 birds facing a genetic wall has become the most carefully managed species recovery in scientific history. But what the DNA revealed is the most remarkable part. The kakapo had already done the hard work, silently. Across 10,000 years, in the forests of a small island at the bottom of the world, they had purged their own damage, they had cleaned their own genome, they had survived their own private extinction long before humans arrived with transmitters and genome sequencers and drones. The scientists who counted 51 birds in 1995 were right to be afraid. But the DNA told a different story. It said, "These birds have survived worse." It said, "They are not as fragile as they look." It said, "Give them a chance, and they will do what they have always done." Somewhere on a predator-free island in New Zealand right now, a fat, green, flightless, nocturnal parrot that smells like honey is booming into the dark. And for the first time in a long time, there is a female listening.